Our today's topic is Calvin cycle or C3 cycle. Photosynthetic carbon dioxide fixation is the point of entry of carbon into organic molecules. Light means sunlight provides the energy, that is solar energy. And in the chloroplast, this solar energy is converted into chemical energy in the form of ATP and NADPH. Calvin cycle is driven by these energy-rich molecules. To synthesize 3 carbon molecules of sugars which act as building blocks for other important carbohydrate molecules like glucose, starch, and cellulose. Now, what is carbon dioxide fixation? When a molecule of carbon dioxide from the environment joins up with another molecule that contains 5 carbons, that is ribulose 1,5-bisphosphate, this process is called carbon fixation. When carbon dioxide joins up with ribulose 1,5-bisphosphate, a chemical compound containing 6 carbon atoms is formed. And this 6 carbon atom compound is subsequently broken down into 2 molecules of a compound containing 3 carbon atoms called 3-phosphoglyceric acid. Right? In the presence of enzyme, ribulose 1,5-bisphosphate carboxylase oxygenase means ribisco. Right? Calvin cycle. Millions of years of evolution have produced significant biochemical variant for fixation of atmospheric carbon dioxide into carbohydrates, namely C3, 3-carbon fixation product, C4, 4-carbon fixation product, CAM pathway means crassulacein acid metabolism pathway, and SAM, submerged aquatic macrophytes. Right? Despite much diversity in life and chemical processes, all of these pathways focus upon a single enzyme, which is by far the most abundant protein on earth, called ribisco. We have already done ribisco in our previous lecture in detail, and ribisco is localized into the stroma part of chloroplast, and this enzyme enables the primary catalytic step in photosynthesis carbon reduction in all green plants and algae, right? But today we will discuss Calvin cycle or C3 cycle. Calvin cycle is also known as Calvin-Benson cycle or the dark reactions. It is a series of biochemical reactions that take place in the stroma of chloroplast during photosynthesis. It was named after the American biochemist Melvin Calvin because he elucidated this pathway in 1950s. Calvin cycle is an essential part of the photosynthetic process in plants, also in algae and some bacteria. Its primary function is to convert carbon dioxide molecule from the environment into organic molecules, right? Which serve as an energy source for the organisms. So, when carbon dioxide molecule from the environment enters the chloroplast and in C3 cycle and joins up with ribulose 1,5-bisphosphate and ATP and NADPH molecule, which are formed from the light reactions, a product 3-phosphoglyceric acid is formed, right? So this is how the Calvin cycle starts. Different steps in Calvin cycle. First step is carbon fixation. You can say here this is a Calvin cycle. 3 steps are there. This is carbon fixation first step. Second is reduction and third is regeneration, right? When molecule of carbon dioxide from environment joins up with ribulose 1,5 bisphosphate in the presence of enzyme rubisco, first 3 carbon unstable intermediate 3 molecules of unstable intermediate 6 carbon compound is formed. Unstable intermediate 6 carbon compound, when react with 3 molecules of water in the presence of same enzyme, 3-phosphoglyceric acid is formed. And this 3-phosphoglyceric acid means 3-PGA is the first stable product of dark reaction of photosynthesis, right? Second step is reduction. Here, 6 molecules of 3-phosphoglyceric acid in the presence of ATP and enzyme kinase converted into 1,3-diphosphoglyceric acid plus ADP are formed. When 6 molecule of this 1,3-diphosphoglyceric acid joins with NADPH2 in the presence of enzyme triose phosphate dehydrogenase, 3-phosphoglyceraldehyde is formed plus NADP plus phosphoric acid is formed, H3PO4, right? Third step is regeneration. Some of the molecules of glyceraldehyde 3-phosphate are used in the production of glucose molecules, while the rest are recycled to regenerate the 5-carbon ribulose 1,5 bisphosphate to receive fresh molecules of carbon for the cycle, Calvin cycle, right? So 3-phosphoglyceraldehyde in the presence of triose phosphate isomerase converted into dihydroxyacetone phosphate, DHAP. 3-phosphoglyceraldehyde first react with DHAP in the presence of enzyme aldolase. Fructose 1,6-diphosphate is formed. This fructose 1,6-diphosphate in the presence of enzyme phosphorylase is converted into fructose 6-phosphate. Some of the molecules of 3-phosphoglyceraldehyde, instead of forming hexose sugar, is diverted to regenerate ribulose 1,5 bisphosphate to receive fresh molecules of carbon, right? Means 3-phosphoglyceraldehyde to ribulose 1,5 bisphosphate. But this 3-phosphoglyceraldehyde react with fructose 6-phosphate in the presence of an enzyme transketolase, erythrose 4-phosphate and xylulose 5-phosphate are formed. Erythrose 4-phosphate then react with DHAP in the presence of enzyme aldolase, and it gives C2-heptulose 1,7-diphosphate. This C2-heptulose 1,7-diphosphate in the presence of ADP and enzyme phosphatase is converted into C2-heptulose 7-phosphate with release of ATP, right? C2-heptulose 7-phosphate plus phosphoglyceraldehyde in the presence of transketolase then will give xylulose 5-phosphate plus ribose 5-phosphate. This ribose 5-phosphate in the presence of ATP and enzyme phosphopentokinase will give ribulose 1,5-diphosphate plus ATP. In the dark reaction, carbon dioxide is fixed to carbohydrates and the carbon dioxide acceptor ribulose diphosphate is regenerated. So in Calvin cycle, 12 NADPH2 and 18 ATPs are required to fix 6 carbon dioxide molecule into one hexose sugar molecule. That is, this hexose sugar molecule is fructose 6-phosphate. So total 6 carbon dioxide plus 12 NADPH plus 18 ATP, and the product is fructose 6-phosphate plus 12 NADP plus 18 ADP and 17 inorganic phosphate molecules. So this is C3 cycle. You can see here with formulas, chemical formulas of different chemical compounds. 3 steps: carbon fixation first, second reduction phase, and third is regeneration phase, right? So this is all about C3 cycle. Thank you.